Today I'm going to introduce my research project supervised by Dr. Sandy Smith. The title is Comparative Study on Biochar Salt Absorption Capacity in Different Saline Concentrated Solutions. This is the outline of my presentation. So first, background information. Soil salinity can increase as a result of cropping, irrigation, and land management. Soils with a high concentration of sodium salts have additional problems such as poor soil structure, poor infiltration or drainage, and limited plant growth. As soil degradation is a global issue, there is currently much research into reclamation and amelioration of saline or sodic soils. Biochar is often promoted as a way of ameliorating degraded soils. Biochar is produced by the pyrolysis of naturally derived organic matter such as wood chips in a low oxygen environment to form high carbon products that can be applied to soils. Biochar has been found to reduce soil stress through sorption, improving soil properties. Salinity is analyzed through electroconductivity measurements. Electroconductivity is a term used to describe a measurement unit of salinity. It allows areas of low and high electrical conductivity to be identified. To understand which variables affect biochar's salt absorption capacity, this research paper focuses on the following specific objectives. First, to measure the salt absorption capacity of biochar through electroconductivity analysis, and second, to evaluate the salt absorption function of different biochar morphologies under different salinity conditions. The hypotheses are: first, saline solutions with biochar will have better salt absorption than solutions without biochar. Second, saline treatments with biochar will decrease electroconductivity values due to improved salt absorption. Based on particle morphology, we use 4 different types of biochar. First is biochar pellets and biochar powder. We can see that biochar pellets are much bigger than biochar powder. The other types are sugar maple biochar fragments and conifer biochar fragments. Also, biochar fragments don't have a specific shape. For example, sugar maple fragments are smaller than conifer fragments. The saline solutions were prepared with 50, 500, 1000, and 2000 parts per million of sodium chloride mixed in 15 milliliters of deionized water and 0.75 grams of biochar. Then we placed the treatments into 25 milliliters glass containers. The solutions were prepared in triplicate because electroconductivity is variable. Also, 2 control groups were prepared. The first control group consists of deionized water with different salt concentrations, and the other was prepared only by mixing deionized water with biochar and no salts. Electroconductivity was measured 1 hour after preparing the samples, and then we placed the samples in a shaker for a week to measure electroconductivity again after that period of time. Statistical data and graphs were analyzed with RStudio. Data for each dependent variable were first analyzed for normal distribution by the Shapiro-Wilkes normality test and for equality of variances among biochar and salinity treatments by the Levine tests. Data meeting these assumptions were further tested on the influence of biochar type and salinity level on the variability of electroconductivity by a two-way analysis of variance tests or ANOVA. Now I'm going to discuss the results of the experiment. The results are discussed as the interaction of biochar on the different saline concentrations treatments over time, and a comparison is made between the 4 different biochar types. For day 1 and 7, a significant relationship is found between biochar and salinity, which is supported by a p-value lower than the common reference value of 0.05. In other words, biochar type and salt concentration are the factors that influence electroconductivity values. However, contrary to our initial hypothesis, biochar electroconductivity measurements did not consistently show a decrease in salinity. Biochar solutions increased their electroconductivity values after a week of being prepared. A finding that supports this result is the increased electroconductivity measured in the control group with pure deionized water and different saline concentrations without biochar addition. Electroconductivity increased by approximately 0.5 to 12% from day 1 to day 7. It is important to note that temperature for day 1 was recorded to be 23 degrees Celsius, whereas temperature for day 7 was 25 degrees Celsius instead. Therefore, we believe that temperature played a major role in rising electroconductivity values, as higher temperatures might evaporate water, leaving sodium ions in the solution behind. We also found that biochar pellets presented a reduction on electroconductivity values at the 50, 500, and 1000 parts per million salinity levels, in contrast to other biochar types. This is because biochar pellets have lower surface area to volume ratio, in contrast to the other biochar types. This characteristic allows biochar pellets to hold more ions on its surface. For biochar powder, the electroconductivity values increased for all salinity levels. This is because biochar powder is characterized by high surface area to volume ratio and has increased porosity that prevents sodium ions to get retained on its surface. Next, sugar maple biochar fragments showed a similar pattern. Electroconductivity values increased at the 500, 1000, and 2000 salinity level, but showed a slight decrease at the 50 parts per million salinity level. And finally, conifer biochar fragments showed a trend similar to sugar maple fragments. Electroconductivity values slightly decreased at the 50 parts per million salinity level, but increased at the other salinity levels. According to these results, the electroconductivity changes related to biochar addition to saline solutions might differ depending on biochar's particle size. For example, a larger particle size, which is the case of pelletized biochar, might increase sodium absorption capacity, whereas a smaller particle size might not. We can state that biochar's particle size, shape, and internal structure likely play important roles in controlling sodium ion storage because they alter pore characteristics. For instance, biochar has pores inside of particles. These pores are known as intrapores and might provide additional space for ion storage beyond the pore space between particles or interpores. As a result, electroconductivity values will decrease. This addition of biochar grains with different shapes and sizes will change the absorption of minerals in the salinity solutions. For instance, fine biochar particles will not absorb much ions in contrast to bigger biochar particles. And these are the conclusions of our project. Electroconductivity of saline solutions is highly influenced by changes in temperature. High temperatures tend to increase electroconductivity values, and also pelletized biochar performed better in low saline treatments. Biochar pellets have a greater potential to absorb more ions due to lower surface area, in contrast to fragmented and powdered biochar. And this is our recommendation. Biochar pellets might be suitable to reduce soil stress in saline soils. This is because lower surface area to volume ratio and also because it has the capacity to retain ions on its surface. And lastly, I would like to thank Dr. Sandy Smith for guiding me through this project, also to Melanie and Dr. Sean Thomas for giving me the chance to work in the lab. Thank you for listening to this presentation. These are the references.